It's a pleasure for me to introduce David Eisner here to give the G.L. Brown Prize Lecture. David studied natural sciences at Cambridge University, and after that he realized he's actually more interested in physiology, so he went on to Oxford, did a PhD in Oxford. After that, went on to UCL, and at UCL he started to work on red blood cells and started to work on cardiac myocytes and generally on the heart. From there, he moved on to Liverpool, worked in Liverpool for quite a while, and in 2000 took up the BHF professorship for cardiac physiology at Manchester University, where he still is today. And yeah, because your lecture is interesting and long, we rather give you the chance to give the lecture now.
Too kind.
Calcium in the heart, from physiology to disease. I'm looking forward to it.
Thank you very much, Katja. That's a very kind introduction. So, I'd like to begin by thanking you at the Open University for kindly inviting me to come here. I'd never been here before, and I've had a very enjoyable day so far, so thank you. I'd also like to thank the Physiological Society for awarding me this travelling lectureship. Now, the lecture is given in honour of G.L. Brown, who was a very distinguished physiologist in the mid-second half of the 20th century. He was head of department and professor of physiology at University College London before moving to Oxford, where he was Waynflete Professor of Physiology, and it's in his memory that this lecture has been established. I want to talk today about the contraction of the heart. This is actually my heart, studied in a magnetic resonance spectrometer, atrium, ventricle, and here you can see the aortic valve opening each time blood is pumped out of the heart. Well, for the rest of the talk, when I talk about the heart, I won't be meaning the whole heart. Everything I do is done at a reductionist level on the isolated cardiac myocyte. So this one is about 120 microns long. My friends will be pleased to know this isn't actually from my heart, this is from a rat's heart. In the rat, it would of course be connected to other ventricular myocytes. You can see where the neighbours have been torn off here and here. And in the attacked heart, it would be electrically excited by its neighbours. In the lab, it's electrically excited by passing current into it, as shown here. And I'll come back to that in a moment. So this is the topics I'm going to touch on in this lecture. I won't bother enumerating them, but it'll just help you to see how far through the lecture we're getting. So I think as a scientist working on the heart, one needs some humility, because long before scientists were interested in the heart, a whole collection of philosophers and artists of various sorts were also interested in the heart. I think the reason they were interested in the heart rather than the brain, for example, is the heart is the only organ in the body that you can tell what it's doing just by touching your chest. You put your hand on your chest, you can feel the heart beating. We don't have the same contact with our kidneys or our brains, for example. And therefore you can find, if you want to start a lecture like this, countless things from the arts dealing with the heart. And one of my favourites is this one. The words are from the poet Müller, the music is from Schubert, and it's from a song cycle called Die Winterreise, A Winter's Journey. And the particular song I'm going to play you a little bit of is called Die Post, The Post. And the singer is waiting for a letter from his loved one, and therefore he's rather anxious. Because he's anxious, his heart is beating rather strongly and slightly irregularly. So don't concentrate so much on the words, just listen to the piano accompaniment, which I think you'll agree is a great lub-dub of a heart. The actual bit I'm going to play you, Was drängst du denn so wunderlich mein Herz? Why do you beat so mysteriously my heart?
Die Post bringt keinen Brief für dich, mein Herz, mein Herz. Was drängst du denn so wunderlich, mein Herz, mein Herz?
So, I think you have to agree that's quite a good heartbeat. The scientific era of the heart, or at least calcium in the heart, starts with Sidney Ringer. Ringer was a clinician at University College Hospital, London, and by his time it was already known that if you took the heart out of a frog, it would continue to beat. And Ringer was interested to know what components was required to make the heart beat. And in 1882, he published a paper saying that all you need for the heart to beat is a simple solution of sodium chloride. Now, if that result had stood the test of time, I wouldn't be here today talking to you on this subject. But fortunately for me, in 1883, he published what I think is one of the most important retractions in the whole of physiology. It's published in the Journal of Physiology, the official journal of the Physiological Society. And the title of the paper is a further contribution regarding the influence of the different constituents of the blood on the contraction of the heart. Now, there are at least a couple of students in the audience here. If you ever submit a paper to a journal of which the first 3 words are a further contribution, you run the risk it won't even be sent out for review, because a further contribution in this age means that it's not sufficiently original. Fortunately for us, and certainly fortunately for Ringer, the paper was sent out for review, and many of you may know what I think is the most important sentence in the whole paper. I discovered that the saline solution which I had used had not been prepared with distilled water, but with pipe water supplied by the New River Water Company. Today, for pipe water, read tap water. Ringer immediately repeated his experiments using distilled water, and when he added sodium chloride in distilled water, the heart stopped beating immediately. And he therefore correctly concluded that there was a contaminant in the pipe water which was required for the heart to beat. He commissioned an analysis of the pipe water and added back the contaminants one by one. And only when he added back the calcium did he find the heart started beating again. And therefore, he discovered that calcium was required for the heart to beat. That's the scientific message, which many of you will know. There are a couple of other things that come out of his little story, one of which is, I think, a major medical and clinical advance. So Ringer discovered not just how much calcium was required to keep the heart happy, but how much of potassium, etc., etc. And therefore, he worked out what is required to keep isolated organs, and indeed the human body, functioning well. And as a result of that, it means that in every hospital in the world, one can safely give patients extracellular fluids because of the fundamental work that Ringer did. And in his memory, these solutions are often known as Ringer's solution or Ringer's lactate. The third message that comes out of it is also really designed for the students in the audience. Look at the English grammar here. I discovered, the active voice, that the saline solution which I had used, again active voice, had not been prepared, passive voice. So what's Ringer saying? He's saying that he was the clever guy who made the discovery, he was the hard-working chap who did the experiment, but clearly here somebody else had made the mistake. So as I always say to people, if you remember nothing else from his lecture, the next time you have to retract a paper, just think about who you're going to blame. So, 130 years later, we know the reason that calcium is required for the heart to contract is because the beating of the heart, the contraction of the isolated cell, is triggered by a rise of calcium. So here we've got an isolated myocyte. We're electrically stimulating it. This one is loaded with a fluorescent indicator, flow 3, which effectively changes color on this picture when calcium rises. So at rest, the image is blue. As calcium rises, it turns to yellow. What I want you to notice is there is a uniform rise of calcium throughout the cytoplasm. And that'll contrast with what I talk about at the end of the lecture. You may be interested to note that in the 2 nuclei in this cell, calcium rises more slowly, but that's not what I'm interested in today. We can quantify this rise of calcium, the so-called systolic calcium transient. At rest, calcium starts off low, to 100 nanomolar, 0.1 micromolar, rises to the order of micromolar during systole, and then relaxes. This is all triggered by the action potential, the electrical signal in the cell. Starts off negative, goes positive, and then repolarizes. This leads to the calcium transient, and after a small amount of time taken for calcium to bind to the contractile proteins, the cell contracts, or the heart contracts, and therefore ejects blood. The other thing to remind you about is the major way the force of contraction of the heart is varied is by changing the size of this calcium transit in the ventricular myocytes. So in exercise, the calcium transient gets bigger. In heart failure, the calcium transient gets smaller. Well, this is what's happening in your cells in your ventricle as you sit listening to me. But as many of you will know, for experimental purposes, rather than relying upon nature's action potential, it's much more convenient to use the voltage clamp technique. So what we do here is make electrical contact with the inside of a cell with this patch pipette. The patch pipette is connected to a feedback amplifier which measures the voltage and then injects just enough current to change the voltage to the desired level. And the clever thing about this technique then is that the amount of current that the amplifier injects is exactly the same as that passing across the cell membrane, so we can measure the movement of ions across the cell membrane. So here we step the voltage from -40 to 0. The current trace here, this downward deflection, is positive ions entering the cell. In this case, this is the opening of calcium channels, the calcium current, the L-type calcium current. The channels open and then they close or inactivate, as shown here. This leads to a calcium transient, and this leads to contraction. So this is one source of calcium, calcium entering from outside the cell via the L-type calcium current. But it's not the only source of calcium, and indeed it's not the major source of calcium. The major source of calcium is from an intracellular store, the sarcoplasmic reticulum, or SR for short. So this is a schematic of the cell. This is the surface membrane. And every sarcomere, so every 2 microns or so, you get these invaginations of the surface membrane, the so-called transverse or T-tubules. And they're required because by mammalian standards, these cells are very large. They're 20 or 30 microns in diameter. And these transverse tubules allow the action potential to propagate into the cell. And many of the channels that we're interested in are represented on these transverse tubules, including the L-type calcium current. So calcium is released from the sarcoplasmic reticulum, and it's released through a special calcium channel known as the Ryanodine receptor. And the Ryanodine receptor has 3 properties that I want to tell you about briefly now. The first is the probability that it's open, and we call that PO, is increased by an increase of cytoplasmic calcium. That's calcium on this side. Secondly, PO is also increased by an increase of calcium inside the sarcoplasmic reticulum. And finally, the open probability is increased experimentally by the drug caffeine. So during the normal cardiac cycle, calcium enters via the L-type calcium channel. The calcium concentration rises here, and because of this property, the Ryanodine receptors open, releasing calcium into the cytoplasm and producing the upstroke of the action potential, and therefore producing cardiac contraction. But if the heart's going to work as a pump, it's not good enough that it can contract. It must also relax. And relaxation requires that calcium be lowered to resting levels. Calcium is lowered in 2 ways. First, it's pumped into the SR by the SR calcium ATPase, called SERCA. Now, SERCA's activity is regulated by 2 factors. One is the availability of calcium, and the other is this accessory protein, phospholamban. Phospholamban normally inhibits SERCA, but when phospholamban is phosphorylated, as occurs during beta-adrenergic stimulation in exercise, this inhibition is overcome and SERCA pumps more quickly. Calcium is also pumped out of the myocyte by sodium-calcium exchange, and this uses the energy provided by 3 sodium ions entering the cell to pump 1 calcium ion out. And because 3 positive charges enter and only 2 leave, this means that this generates an electric current. And today that's important for 2 reasons. One, we can use this electric current to measure the activity of sodium-calcium exchange. And secondly, as I'll show at the end of the lecture, this can lead to some ventricular arrhythmias. The other thing to emphasize is the events I've talked about happen each time your heart beats. And in the steady state, therefore, calcium must be in flux balance. That is to say, influx must equal efflux. The calcium entering here must equal that pumped out there. The amount released here must equal that taken back up here. And really much of what I'm going to talk about for the rest of the lecture comes down to this steady state flux balance. In this respect, the heart is a bit different from many other cells. If you think about, for example, vascular smooth muscle, calcium rises over seconds, tens of seconds, and then relaxes slowly. The heart is very different. In the steady state, on each beat, that amount of calcium that comes in must equal that goes out. And I can demonstrate to you that the heart is in calcium flux balance, and also use this slide to show the experimental approach we take. These traces you've seen already. This is the voltage step, the systolic calcium transient, and here's the membrane current. We wanted to know how much calcium enters the cell and how much leaves. Well, this is current, and this is time, And if we multiply the two together, we get charge. So the multiple of the two together is shown by the red area here. So on this calcium current, the L-type calcium current, this is the amount of calcium that enters. The running integral is shown down below. And on this one pulse, somewhere between 3 and 4 micromoles of calcium enter per each litre ventricular cell volume. When calcium is pumped out here, it's done largely by the sodium-calcium exchange. This current here is the sodium-calcium exchange current. If I draw the baseline there, you can see the current relaxing to this baseline. It is a bit small, so I've amplified it here. This is the sodium-calcium exchange current. We can integrate this again, and this area here shows you how much calcium is pumped out. And you can see here the amount of calcium pumped out equals exactly the amount pumped in. So calcium entry equals exit. People sometimes think that the integral here looks a lot smaller than that there. But you have to remember that when calcium enters on the calcium current, it carries 2 positive charges. But each time a calcium ion is pumped out on the exchanger, only 1 positive charge net moves. And therefore, you mentally have to multiply this by 2. So the cell is in calcium flux balance. So you've seen this scheme already. What we want to know is which of these steps are controlled to alter the force of contraction of the heart. And there are 3 obvious candidates. The L-type calcium current, the bigger this is, the more calcium it enters, and therefore the more Ryanodine receptors may get opened. The properties of the Ryanodine receptor, you might expect that the more open, the more calcium will be released. And finally, the amount of calcium in the sarcoplasmic reticulum will also determine how much is released. I'm going to talk today mainly about the calcium content of the SR. I'll talk a little bit about the Ryanodine receptor, and I haven't got time to cover the calcium current today, but it is also quite important. So I thought I might start by showing you what happens in perhaps the best known positive inotropic maneuver. That is to say, the maneuver that increases the force of contraction of the heart. And this is the one that occurs every time you exercise. So these are the positive inotropic effects of beta-adrenergic stimulation. This is recorded in an isolated ventricular myocyte under basal conditions. You can see the calcium transients here in the cytoplasm. We've got another calcium indicator in the cell here, measuring calcium in the sarcoplasmic reticulum. And you can see each time calcium rises in the cytoplasm, it falls in the sarcoplasmic reticulum. And since you'd expect that, I hope, because the major source of this calcium is calcium being released from the SR. What we then do is to beta-adrenergically stimulate by adding the synthetic catecholamine isoprenaline. And when we do that, as has been shown by many before, the calcium transients get bigger. If you look at what's happening in the sarcoplasmic reticulum, the sarcoplasmic reticulum calcium content here has got higher. The depletion of calcium in the SR has got much bigger. And as I said, the calcium transient's bigger. So what's happening here is mainly governed by 2 things. Phospholamban is phosphorylated by beta-adrenergic stimulation, so circa pumps more quickly. This leads to this increase of calcium in the sarcoplasmic reticulum and makes more calcium available for release, leading to a bigger calcium transient. In addition, phosphorylation of the L-type calcium channel increases it and increases the triggering of release, so you get more release due to a bigger depletion of the SR. So those are the 2 primary things that account for this increase of the calcium transient. In a few minutes, I'll discuss whether the Ryanodine receptor is also involved in this. It wouldn't be fair, though, to finish without talking about another physiologically important effect of catecholamines, and this is the so-called positive lusitropic effect. I think you can see up here, not only are these calcium transients bigger than these, they also decay more quickly. That's emphasized here. The control baseline is in blue. In isoprenaline, it's in red, and you can see this decays much more quickly. This is physiologically vital. Under normal conditions, when you beta stimulate, not only does your ventricle beat more strongly, but the heart rate goes up. So there's less time in diastole for the heart to refill with blood. And the heart compensates for this by making systole last much less time by a faster reuptake of calcium. And this faster reuptake of calcium is again due to phosphorylation of phospholamban, leading to increased circuit activity. You don't see it here because we're experimentally just using stimuli applied at a constant rate. Okay, so the first question I want to address in the talk proper is the link between the sarcoplasmic reticulum calcium content and the size of the calcium transient. I've already shown you that we can measure the SR calcium content. We can change it experimentally and ask what happens to the size of the calcium transient. And when we do that, as shown here, you get this sort of relationship. It's much steeper than linear. You can fit it with a power function, and on average, the size of the calcium transient is proportional to the cube, the third power of SR calcium content. Well, this steep relationship comes, I think, from several factors. One is the fact that as you increase calcium in the SR, the driving force for calcium to leave increases. But also, as I've told you, as you increase calcium in the SR, more Ryanodine receptors open. Whatever its origin, I think this steep relationship is physiologically very important. So what are the consequences of it? Well, the first one is that even modest changes of SR calcium content, because of the steepness of this relationship, will have big effects on the amplitude of the calcium transient. So, for example, if you increase heart rate, this will lead to an increase of SR calcium content, and because of the steepness of this relationship, will lead to a useful increase of contractility. But it also relates to heart failure. It's been known for many years that the main reason the failing heart beats more weakly is because the systolic calcium transient becomes smaller. And it's been thought that this is due to a decrease of SR calcium content. So my colleagues were interested in studying this. This is a model, a ferret model of heart failure. Calcium transients from a control cell, from an animal in heart failure. You can see the calcium transient is smaller in failure. If you look at the mean data, the calcium transient in failure is 0.47, just under half of the control level. What they wanted to know was this due to a decrease of SR calcium content? They measured the SR content, and in failure, the SR content decreased, but only to 0.75 of the control. So can this account for the decreased contractility? Well, yes, it can. I've told you that the calcium transient goes as the cube of SR content. So the calcium transient, if you'd expect 0.75 cubed, which is 0.42, and that's in pretty good agreement with the observed 0.47. So don't get too hung up on the numbers, but what this means is that even modest decreases of SR calcium in heart failure can have big effects on the calcium transient and thence on contractility. The final importance of this steep relationship is that, to be teleological for a moment, it presents the cell with a real challenge. If the cell wants to maintain the amplitude of the calcium transient constant from beat to beat, then because of this steep relationship, it has to control the SR calcium content incredibly precisely, because even a small unwanted change of SR calcium will result in a big change of the calcium transient. So therefore, how does the cell control its SR calcium content? Well, if you go to textbooks, you'll see much discussion about the fact that phosphorylation of phospholamban by these two kinases regulates circuit activity. Now, I've no doubt that this is true, and I've no doubt this will change SR content, but it's not what we think is the most important thing for beat-to-beat regulation of SR calcium. Rather, we think it's something that we've called autoregulation, whereby the calcium released from the sarcoplasmic reticulum affects movements of calcium across the surface membrane, the sarcolemma, and thence affects the SR content. I want to show you one experimental study that led us to this conclusion. So, as in many cases in physiology, you're presented with a physiological system that works beautifully. The heart does an amazing job of controlling its SR calcium content. And if you want to understand how it does it, it's often very useful to disable the system and see how the system then responds. So we've disabled it by emptying the SR of calcium. Again, we're measuring cytoplasmic calcium and stimulating here, these are systolic calcium transients. We then apply a high concentration 10 millimolar caffeine. Caffeine binds to the ryanodine receptors, makes them open, releases calcium into the cytoplasm. This calcium is then pumped out of a cell by the sodium-calcium exchange, and a few seconds later, you have a cell with no calcium in its SR and the normal calcium in the cytoplasm. We wash the caffeine off and start stimulating. The first calcium transient is very small because the SR is empty. This calcium transient is presumably due to calcium coming from outside the cell. But within 10 or 20 seconds, the calcium transient has come back to control levels, and presumably, therefore, the SR is full. So what I want to do now is to look at this area highlighted here and ask the question, how does the SR refill from here to here? And that's shown here. So we start off, we've emptied the sarcoplasmic reticulum, the SR is empty here. 20 seconds later, the SR is completely full. We go from a small transient to a big one. To understand what's happening, I'm going to have to show you in more detail the membrane currents accompanying first the steady state calcium transient and then this small one here. So the steady state calcium transient is shown here. Here's the calcium transient. Here's the membrane current. The calcium enters on the L-type current, leaves on the exchanger. When the SR is empty, we've got the smaller calcium transient, and here's the current. So now let's go even more magnified in time and look at the calcium entry on the L-type current. When the SR is full, our calcium current switches on and then inactivates or switches off. I'm now going to superimpose on it the calcium current when the SR is empty here. And you can see the activation is the same, but this inactivation or turn-off is much slower. So therefore, much more calcium enters on the red trace than on the blue one. The explanation for that was known from work before ours. This inactivation of the L-type calcium current is a calcium-dependent process. So the bigger the systolic calcium transient, the more complete and faster the inactivation, the turn-off of the L-type calcium current, and therefore less calcium enters. So less calcium enters here than enters with the smaller calcium transient here. What about the efflux of calcium from the cell? This is the sodium-calcium exchange pumping calcium out when the SR is full, a big current. When the SR is empty, the current trace is much smaller. This is even simpler to understand. The sodium-calcium exchange is activated by calcium. The bigger the calcium transient, the more calcium is pumped out on the exchanger. So what we've got now are two membrane currents that are affected by the size of a systolic calcium transient. The calcium, as the calcium transient gets bigger, the calcium entry decreases and the calcium exit or efflux increases. Why is this important? This you've seen already. Calcium enters, the ryanodine receptors open, and we get the calcium current. Now, obviously, the main function of this calcium transient, the main function of the calcium transient is to activate contraction. But there are other targets for calcium in the cell, many other targets, and two of them I've just told you about. One target is the L-type channel. This calcium transient inactivates it and decreases calcium entry. Another one is the sodium-calcium exchange. The calcium transient activates it and increases calcium efflux. So imagine that we have an increase of SR calcium content. As I've told you, this will increase the size of the calcium transient. This will decrease calcium entry, and it will increase calcium exit. And the combination of decreased calcium entry and increased calcium exit will decrease the calcium content of the cell, and therefore it will decrease the calcium content of the SR back to control levels. So we think this very simple negative feedback system is how this cell regulates its calcium. Changes of SR calcium affect the calcium transient. These, in turn, affect fluxes of calcium across the surface membrane and restore calcium back to steady levels. So I think it's just an incredibly simple feedback mechanism. There are two other things I want to say about this. The first one is many of you will know about store-operated calcium entry. So in many cells, particularly non-excitable cells, the endoplasmic reticulum, as it is, when it loses calcium, when it's empty of calcium, sends a signal out to activate special calcium, store-operated calcium entry mechanisms in the surface membrane of the cell, leading to a replenishment of calcium in the store. Now, I don't think the adult cardiac myocyte makes use of these store-operated channels, but what this feedback mechanism is doing here is achieving exactly the same result. When the store empties, it sends out a signal, here a decrease of the calcium transient, to affect fluxes of calcium across the surface membrane. You might ask, why does the heart not use store-operated channels? All I can do in this context is to speculate. The fluxes of calcium across a cardiac cell membrane are of the order of 100 times greater than those in the typical non-excitable cell. And if the heart wanted to do it with store-operated channels, it would have to have a lot of store-operated channels in addition to these pathways. It may be energetically, therefore, much more efficient just to modify these pathways which it needs for its normal contractile business. But that is just speculation. The other thing I want to point out is people sometimes think I'm saying that because of this feedback mechanism, it's impossible to change the SR calcium content. Well, nothing could be further from the truth. This mechanism sets the SR content at a set point, steady from beat to beat. But the actual value of that set point depends upon the expression and activity of all these calcium-handling proteins, the L-type channel, the Ryanodine receptor, sodium-calcium exchange, and SERCA. And if you change any one of those, you'll change the set point reached. And a good example is provided by heart failure. I've already told you that in heart failure, the sarcoplasmic reticulum calcium content decreases. And there are at least 3 reasons by which this occurs. Firstly, you get an increase of sodium-calcium exchange activity, so more calcium is pumped out of a cell, less is available to the SR. The Ryanodine receptors become leaky, so the SR finds it harder to hang on to calcium. And finally, SERCA activity decreases and less calcium is taken back into the SR. As a result of all these 3, you reset the set point and you get a decrease of the sarcoplasmic reticulum calcium content. So this feedback mechanism can also rationalize what's happening in heart failure. Now in this lecture, I'm going to talk a lot about calcium fluxes and somewhat about heart failure. I don't want you to go away thinking the only thing that happens in heart failure is changes in calcium fluxes. There are massive changes in almost every property of the cell, and one of them is very easy to see in the structure of the cell. This is a membrane-staining dye on actually a sheep atrial myocyte. This is the surface membrane, but these lines here are the transverse tubules I mentioned before. There are lots of them here. When we take these sheep and induce heart failure, this is what the cell looks like. Every single transverse tubule disappears. And therefore, a lot of what happens in heart failure is due to the fact that you've lost all this membrane, which is required to efficiently trigger calcium signaling. So this is just yet another thing that happens in heart failure. There are many other things as well, which I haven't got time to go into today. I want to come back again to this negative feedback control of SR calcium content by addressing this question, which is, what are the consequences if you modify the opening of the Ryanodine receptor on systolic calcium and thence on contraction of the heart? This diagram you've seen many times. I've talked to you so far about the Ryanodine receptor being controlled by calcium, but there are many other things that control the Ryanodine receptor. It has many phosphorylation sites on it. When you phosphorylate the Ryanodine receptor, you increase its opening probability. The intracellular messenger cyclic ADP ribose also increases the open probability of the Ryanodine receptor. And finally, free radicals increase the open probability of the Ryanodine receptor. And it's being suggested that all these actions here will lead to more Ryanodine receptors opening and more calcium being released, and therefore a bigger calcium transient. And in particular, it's been suggested that phosphorylation of the Ryanodine receptor contributes to the positive inotropic effects of beta-adrenergic stimulation. I told you before that the consensus view is that beta-adrenergic stimulation works by phosphorylating the L-type channel and phospholamban. But there are many distinguished people who think that it also works by phosphorylating the ryanodine receptor. So we wanted to investigate this. It's hard just to phosphorylate the ryanodine receptor without also phosphorylating the other targets. So we wanted another way to modify the open probability of the ryanodine receptor. And what we've done is to use low concentrations of caffeine. At low concentrations, caffeine sensitizes the ryanodine receptor to being activated by calcium. So more ryanodine receptors open. And therefore, we expected that if we did that, we would get an increase. If we increase the number of ryanodine receptors that open, we get an increase of systolic calcium. So what does happen? That's shown here. This is the baseline condition. We've got, under the conditions of this experiment, small calcium transients, small amplitude. We then add a low concentration of caffeine. This makes more ryanodine receptors open, so the immediate result is a massive increase in the size of the calcium transient. What I want you to notice, and here's the time bar below, is this isn't maintained. The calcium transients get smaller again, and in the steady state, reached just a few seconds later, the calcium transient amplitude is identical to the control. Now, it doesn't matter how much I enlarge this. These calcium transients are identical in amplitude and time course to the control ones. So we had a paradox. Caffeine has maintained effects on the ryanodine receptor opening if you study isolated ryanodine receptors, but it has only a phasic effect on the amplitude of the calcium transient. What's responsible for this paradox? Well, I'm going to suggest to you, it's this negative feedback regulation of SR content. So, these are the calcium fluxes on each beat accompanying the transients above. The blue is the calcium influx, the red is the efflux. And down below here, we've calculated now the SR calcium content. When we start off, influx equals efflux, the points lie on top of each other, we're in a steady state. SR content is fixed. When we add caffeine, the calcium transient gets enormously bigger, activates the sodium-calcium exchange, which therefore efflux gets much bigger. So now efflux is much bigger than influx. We're no longer in a steady state. The SR calcium content falls. As it falls, the calcium transient gets smaller. The efflux falls, but it's still bigger than influx, until we reach a new steady state here, where efflux again equals influx at a lower SR content. So we've reached a new steady state. The ryanodine receptor is still potentiated by caffeine, but because there's less calcium in the SR, the calcium transient is smaller. So what's happening then? We began by increasing the opening of the ryanodine receptor. This increases the calcium transient, increases calcium efflux. We're no longer in steady state. The SR calcium content falls, and because it falls, the calcium transient falls back to steady levels. So again, this is this negative feedback, which is stopping the perturbation produced by increasing the ryanodine receptor opening. So our conclusion is that if you're looking for a target, let's say you're a drug company, to increase the force of contraction of the heart, forget about making the ryanodine receptor open more, because as I've just shown you, other factors mean that the calcium transient in the steady state is exactly at control level. Okay, I want to spend the last few minutes of a talk talking about the link between leak of calcium through ryanodine receptors and cardiac arrhythmias. Many of you will know that death from heart failure is a major cause of death in the Western world. Of those people who die from heart failure, they die in two ways. Half the people die rather slowly as the heart becomes more and more feeble and less capable of pumping blood. The others die suddenly, they drop down dead, sudden death. And sudden death is death by arrhythmias. The other thing is that many companies over the years have tried to produce drugs, so-called positive inotropic agents, to increase the force of contraction of the failing heart. These drugs work by increasing systolic calcium, presumably by increasing the SR calcium content, but almost without exception, these drugs have been discontinued because they increase deaths from arrhythmias. And what many labs in the world, including those at the Open University here and our own, are interested in is what is the link between calcium, sarcoplasmic reticulum calcium and cardiac arrhythmias. And I think most people would agree that a major link is shown in this diagram here. So now I'm showing you not just one sarcomere, but 3. Under normal conditions, calcium enters from outside, triggers the opening of the Ryanodine receptor. But what we know now is that if you increase the amount of calcium in the sarcoplasmic reticulum, calcium leaks out spontaneously, so-called calcium sparks. Now, the calcium will leak out, some of it will be taken back up, some will be pumped out. But if enough leaks out, it will diffuse ahead to the next region of the sarcoplasmic reticulum, bind to the Ryanodine receptor, make it open, releasing more calcium. Calcium will diffuse ahead again, and so on and so on. And what you'd expect you'd get is a propagating wave of calcium-induced calcium release. And that's exactly what you see here. Again, we're measuring calcium. This cell is not electrically stimulated. It's got so much calcium in its SR, calcium leaks out here and propagates as this wave of calcium-induced calcium release, completely different from the synchronous release that I showed you at the start of the talk. The probability that these calcium waves occur is a very steep, almost a threshold function of SR calcium content. This is the SR content. Here's the frequencies of occurrence of these waves. Below a certain threshold level of SR calcium, you never get a wave. Above this threshold, you get an increasing frequency of these waves. I'll come back to that in a moment. Well, I think you can see that these waves interfere with normal calcium signaling. It's much worse than that. These waves occur during diastole, and diastole in the heart, the cell should be electrically silent. The problem is some of the calcium in the wave is pumped out of the cell by the sodium-calcium exchange. The sodium-calcium exchange generates an electrical depolarizing current, and the consequences of this were noted by Ferrier and colleagues long before the mechanism was established. So Ferrier et al. Were interested in the arrhythmogenic effects of digitalis glycosides. Digitalis glycosides, originally from the foxglove, are given to increase the force of contraction of the failing heart. But the problem is that it's very easy to give too much and produce a whole collection of side effects, including ventricular arrhythmias. So they studied dogs which were given an overdose of digitalis. This is from a canine cardiac Purkinje fiber then. They stimulated the fiber 6 times as shown here, and following the last stimulus, you get this abnormal delayed after depolarization, which you would never see in a normal Purkinje fiber. If they stimulated the same fiber slightly more quickly, now twice a second, the delayed after depolarization here is big enough to excite a train of 3 action potentials. Now, in the context of the intact heart, these action potentials would be extrasystoles, and this is the first stage of producing ventricular arrhythmias. So I think then this is the link from calcium waves to arrhythmias. So the idea then is that if calcium leaks out of the sarcoplasmic reticulum in diastole, it sets up calcium waves. This was originally noted under conditions when an increase of SR calcium content leads to extra leak and therefore leads to waves. I've told you it's important for digitalis toxicity. Subsequently, it was realized that it's responsible for the arrhythmias you get when you reperfuse, when you remove a block in a coronary following ischemia. And it also bedevils the use of inotropic agents. There's also today a lot of interest in the idea that you can get the calcium leak and the calcium waves not simply by increasing the SR calcium content, but by changing the properties of the Ryanodine receptor such that they become leaky. This is thought to contribute to heart failure, arrhythmias in heart failure, and it's also important, and I'm going to talk a little bit about it, in an inherited arrhythmia syndrome called CPVT. What is CPVT? CPVT stands for catecholinergic polymorphic ventricular tachycardia. The polymorphic ventricular tachycardia is just a description of this ECG before it degenerates into ventricular fibrillation. The catecholinergic is due to the fact that the people who present with this arrhythmia are fine and basal conditions, but if they exercise or are otherwise stressed and their catecholamine levels increased, that's when they get their arrhythmia. Half the people who have this arrhythmia have single amino acid mutations in their ryanodine receptor, which make the ryanodine receptors leaky. Normally when they present, they're children, they have completely structurally normal hearts, normal ECGs, until they exercise and they can drop down dead from fatal arrhythmias. So we're interested then in CPVT, and interested in particular in two questions. The first question is, why do leaky ryanodine receptors produce arrhythmias? And I've given you a hint already. And secondly, what's the mechanism by which beta-adrenergic stimulation due to exercise results in these arrhythmias? And I want to touch on these two questions. So the first question we wanted to address was, if you only make the ryanodine receptor leaky, does this produce arrhythmogenic calcium waves? And the way we're doing that, again, is with low concentrations of caffeine. You've seen data like this already. This is the probability of getting a wave as a function of SR content. Below a threshold, you never see a wave. Above the threshold, you always get waves. This is the basal condition. If we add low concentrations of caffeine, the threshold shifts to much lower levels because we've made the ryanodine receptors leaky. Normal SR content is about here, thus explaining why you don't get waves under normal conditions, but we might expect to get waves in the presence of caffeine. So the first question was, if we lower this threshold by making the ryanodine receptors leaky with caffeine, do we see waves? And the answer surprised us initially. These are calcium measurements. We stimulate here. Under basal conditions, you just see systolic calcium transients, nothing in diastole. When we make the ryanodine receptor leaky with caffeine, we see waves. There are 3 waves here. These are the rises of calcium, not due to the stimuli. But what I want you to notice is the waves disappear. There's no waves seen here, no waves at all in the steady state. And the explanation for that seems to be, again, it's this feedback mechanism. This is the calculated change of SR calcium content. These calcium waves, the calcium is pumped out of the cell, so the SR loses some of its calcium. I've told you that increasing SR content produces waves, so it shouldn't come as any surprise to you that lowering SR calcium content abolishes the waves. So let's go back to the children with CPVT. They have mutant ryanodine receptors throughout life, but only when they exercise or are stressed do they get their arrhythmias. So perhaps in hindsight we were doing the wrong experiment here. We were just making the ryanodine receptors leaky. We weren't beta stimulating. So let's repeat the experiment and ask what happens if we increase the opening of the ryanodine receptor in beta adrenergic stimulation. Do we get calcium waves in the steady state? So this is the same experiment now, but now in the presence of isoproterenol, the beta agonist. No calcium waves here. When we make the ryanodine receptors leaky, we get calcium waves, and they persist. This is the systolic calcium transient. This is the calcium wave, systolic calcium transient, calcium wave. So we can mimic the clinical phenotype of CPVT. Just as the children only get their arrhythmias when they both have leaky ryanodine receptors and are beta stimulated, we only get the calcium waves when we have both leaky ryanodine receptors and beta stimulation. So the final question here then is, what's the mechanism by which beta adrenergic stimulation produces calcium waves? And there are 2 in the literature. The one I'm going to try and persuade you is the correct one is that beta adrenergic stimulation, as we've seen already, makes circa pump more quickly and therefore increases the SR content. The other explanation that abounds in the literature is phosphorylating the ryanodine receptor during beta adrenergic stimulation lowers the threshold for release even more. Evidence in favor of the first one comes from this sort of experiment where we measure the SR calcium content. This is without beta stimulation. In beta stimulation, the SR content is much greater, and other data we have shows that this rise of calcium here is enough to turn it from a situation with no waves to a situation here with lots of waves. So we think, therefore, that beta stimulation produces calcium waves by increasing the amount of calcium in the SR. So in other words, whether or not you get a calcium wave depends upon the relationship between the SR calcium content and the threshold required to get a wave. Under basal conditions, the content is less than the threshold and you don't get a wave. When we add caffeine to make the ryanodine receptors leaky, yes, we lower the threshold to less than the initial content, but as I've shown you, the content falls further to less than the threshold and we don't get a wave. If we add modest amounts of isoprenaline to beta stimulate, we raise the content, but not as far as the threshold, and we don't get a wave. Only if we both make the ryanodine receptor leaky with caffeine and beta stimulate is the content now greater than the threshold, and we do get a wave. Now, you're a very polite audience, but I'm sure you're not interested really in the effects, interactions between caffeine and beta stimulation. But let's just imagine that this is a good model of what's happening in CPVT. So if you accept that, we can now understand, if you're born with a mutant ryanodine receptor, why you get your arrhythmia when you exercise, because only now is your SR content greater than your threshold. But that is just speculation. What happens in people with CPVT? For obvious reasons, we can't do it on people, but through our collaborator Silvio Priori, we have access to mice with the mutant ryanodine receptor knocked in. And we can measure the threshold calcium content required to get waves. In the wild-type littermates, you have to raise SR calcium to here to get a wave. In the CPVT heterozygote, only to here. And this is significantly less than this level here. So therefore, indeed, the caffeine experiment was a reasonable one to have done originally. In CPVT, the leaky ryanodine receptor lowers the threshold to get a calcium wave.
Why do I yield to that suggestion, whose horrid image doth unfix my hair and make my seated heart knock at my ribs against the use of nature?
I'll embarrass you all by asking you which Shakespeare play this is from. You're absolutely right. It's Macbeth. If you remember Macbeth and his wife had committed regicide. They'd killed the king. In the modern vernacular, they're therefore very stressed. Their catecholamine levels are therefore very high. And they noticed that what this does is to make their heart, which should normally be seated in the chest, knock at their ribs. Now, I can't pretend this is the first demonstration of CPVT, but I think it's quite a nice account of what happens, what you feel when your heart beats more strongly. For obvious reasons, I'd rather you ignore the doth unfix my hair. So that's Macbeth. Finally, then, I've talked about the problems produced by calcium-dependent arrhythmias. How can physicians treat them? The real problem is the calcium arrhythmias, dependent arrhythmias, are produced by calcium waves, by calcium being released through the ryanodine receptor. Now, you could block ryanodine receptors, but the normal heartbeat depends upon calcium being released through ryanodine receptors. So how can you get rid of the waves without getting rid of the normal heartbeat? So we wondered whether we could use the local anesthetic tetracaine to do this. Now, tetracaine doesn't block ryanodine receptors. What it does is to decrease the probability that they're open. What tetracaine does is to increase the threshold to get a calcium wave. It does the opposite of caffeine. And that's what's done in this experiment here. These are control cells, calcium transients. When we massively beta stimulate, this is much higher concentration than we used before, the calcium transients get bigger, but we get calcium waves, as shown here. And under these conditions, they do persist. What we then did was to keep the beta stimulation with ISO present, but now add on top tetracaine. And you can see the calcium waves disappear completely, and as shown by the dashed line, the calcium transients get bigger. So the calcium waves disappearing is because the threshold for calcium release has increased. The calcium transients get bigger. We think these calcium waves inhibit the subsequent release of calcium. So if you block the waves, the calcium release gets bigger here. At first sight, this seems very attractive. Imagine you have a patient with heart failure. Not only have you cured the arrhythmias by blocking the calcium waves, but you've also made the heart beat more strongly, as shown here. The problem with tetracaine is, yes, it blocks the ryanodine receptor, or rather, it decreases the opening of the ryanodine receptor. It's a local anesthetic. It blocks sodium channels, and at the concentrations required to do this, you'd lose action potentials essentially in all your nerves and in all your skeletal muscle. So you can't use tetracaine. And I always used to end my talks on this very negative way until Bjorn Nolman in the States used not tetracaine, but he used flecainide, a related local anesthetic. And he showed not only did this abolish CPVT in mice, but much more importantly, in humans who had CPVT which was refractory to other treatments. And if anybody's interested, I recommend this paper. I think it's a very beautiful translational paper. So this is what I've talked about in the lecture. Or this is what I won't go through it in any detail. I just want to end up with acknowledgements to people in my lab. So you may not have realized in the past when you've listened to Peter Sellers and Sophie Loren singing this, this is, of course, shows you how much work we have to do now on the heart still, because clearly that cardiologist didn't understand at all what the cause of his patient's arrhythmia was. Thank you for your attention.